Part 4, The Genesis and Diversity. Chapter 16, The Genesis of Life, Introduction. The process of formation is evolution, and the rest is existence until extinction or further evolution happens. The genesis of anything includes how it came into existence, evolution, and continues to exist, existence. We shall recollect the laws of existence before going further into the chapter. The stable will exist, and the rest will perish. Stability is the function of forces of formation, which should be greater than the forces of deformation. Over time, those entities that could resist, reduce, reverse, or replace will exist, while the rest will evolve or go extinct. These laws operate on every entity that ever existed in the universe, accounting for its environment. With regard to life, the laws of existence are applicable from abiogenesis, the first of the primordial replicator. The origin of life is the most interesting and intriguing topic, followed by the mind and origin of the universe. Most cultures and mythologies probably had versions of the origin of life, but religions propose the best recorded ones. God created the universe, impregnated it with life, and handed it to humans, a creation in God's own form. That is it. Nevertheless, individuals often question these theories or prophesy and speculate on how else life could come into existence. Several scientists and philosophers propose diverse ways. Ancient views on the origin of life were deeply rooted in mythology, philosophy, and spirituality. Many cultures attributed life's emergence to divine or supernatural forces. In Greek philosophy, thinkers like Anaximander proposed naturalistic ideas, suggesting life arose from moisture, while Empedocles theorized about elemental combinations. Indian and Chinese philosophies emphasize cosmic unity and cyclical creation, linking life to universal energy. Religious texts, such as the Bible and Quran, describe creation as an act of God. These perspectives often intertwined moral, spiritual, and cosmological interpretations, reflecting humanity's early attempts to understand life's mysteries before scientific explanations emerged. We would never know with absolute certainty about the origin of life in the universe, which is no longer how it was billions of years ago, during the Prebiotic era. We can speculate on the origins based on the properties of the living organisms in the current day. The most accepted theory in modern scientific literature regarding the origin of life is abiogenesis, the process by which life arose from non-living matter. According to this theory, life emerged through a series of chemical reactions on the early Earth, driven by various energy sources such as volcanic activity, lightning, and ultraviolet radiation from the sun. The theory suggests that simple organic molecules like amino acids and nucleotides formed in the primordial soup, a mixture of water, gases, and minerals, and underwent chemical evolution. Over time, these molecules assembled into more complex structures, including self-replicating molecules, likely RNA, which served as both a genetic material and a catalyst. This is known as the RNA world hypothesis. The transition from simple molecules to the first living organisms involved the development of cell membranes, which allowed for the formation of protocells, primitive, cell-like structures capable of maintaining internal environments separate from the external world. These protocells may have led to the first forms of life. In this chapter, I present my opinions, which may or may not align with the scientific literature. I base all my views on existence as the center, emphasizing iterations that favor entities with enhanced existence. This reflects natural selection at the molecular level. Existence is defined by the ability to persist despite environmental conditions. An entity with its material structure and information must be stable over time. No entity with a higher thermodynamic state relative to its environment can continue to exist. Yet, some entities persist due to a unique inherent ability called life. This ability has developed gradually over billions of iterations of formation and deformation. Back to Darwin's warm little pond. My dear Hooker, it is often said that all the conditions for the first production of a living organism are now present, which could ever have been present. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc., present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present day such matter but be instantly devoured or absorbed, which would not have been the case before living creatures were formed. Charles Darwin. Darwin's thoughts on the origin of life, as expressed in a private letter to Joseph Dalton Hooker, offer a glimpse into his speculative ideas on this profound question. In a letter dated February 1, 1871, Darwin proposed a hypothesis that reflects his contemplation on the conditions necessary for life to emerge. The core of Darwin's hypothesis revolves around the notion that the early Earth possessed the necessary conditions for the chemical formation of organic compounds, potentially leading to the emergence of primitive life forms. He described a hypothetical scenario involving a warm little pond containing essential elements such as ammonia, phosphoric salts, light, heat, and electricity. Within this primordial environment, Darwin speculated that a protein compound could have formed through chemical processes, setting the stage for further complex transformations. Darwin's recognition of the speculative nature of his hypothesis is evident in his use of the phrase, "Oh, what a big if," which emphasizes the uncertainty surrounding the process. He acknowledged the immense complexity involved in understanding the origin of life and the challenges inherent in conceptualizing such a significant event. Grasping the origin of life requires interdisciplinary expertise spanning multiple scientific fields, including physics, chemistry, mathematics, and geology. Applying systems thinking and evolutionary principles is essential to comprehend the intricate interplay of factors that may have contributed to the emergence of life. While the specifics of life's origin remain elusive, Darwin's reflections highlight the crucial role of scientific inquiry in unraveling this enigma. Science defines the boundaries of what is possible, stable, and subject to change, guiding our exploration of life's origins and the subsequent evolutionary processes that have shaped Earth's diverse living organisms. It is important to recognize that while the origin of life is an improbable event, once it is initiated, the subsequent evolution of life unfolds through a complex yet spontaneous process driven by inherent mechanisms of change. Thus, although the genesis of life may remain a tantalizing mystery, the ongoing quest for understanding embodies the essence of scientific inquiry and exploration. In my view, Darwin's primordial soup theory offers a more plausible scenario for the origin of life, emphasizing the need for concentrated organic molecules to enable chemical reactions necessary for life. In shallow, warm water environments, these molecules could accumulate in localized regions, driven by natural processes such as evaporation, temperature gradients, and mixing, fostering conditions ideal for polymerization and self-replication. In contrast, deep-sea vents, while rich in chemical compounds, pose challenges for concentration as currents could disperse the molecules, hindering the formation of complex structures. The primordial soup, with its potential for molecule concentration, provides a more favorable setting for life's emergence. Essentials for the origin of life. At the heart of Genesis lie 5 critical elements: carbon, energy, periodicity, water, and time. Each of these factors plays a unique and indispensable role in fostering the conditions necessary for the emergence and evolution of life. Their interplay creates a dynamic environment within which the complexity of life can develop. The origin of life is contingent upon several essential factors that interact and shape the complexity of living systems. Carbon stands as the backbone of life's molecular architecture. Its ability to form stable covalent bonds with itself and other elements enables the construction of diverse, intricate molecules like proteins, nucleic acids, and carbohydrates. These molecules are central to life's structural integrity, enzymatic functions, and genetic information storage and transfer. Carbon's unique ability to catalyze polymerization is indispensable for self-replication and evolution. See Chapter 10. Energy is another key component that drives the biochemical reactions necessary for survival and growth. Early Earth's energy sources, such as sunlight and geothermal heat from hydrothermal vents, facilitated the formation of organic molecules. These energy sources power essential processes such as metabolism and molecular synthesis. Periodicity, the cyclical changes in environmental conditions, provided a stable yet dynamic framework for molecular evolution. The regular alternation of light and dark and seasonal changes influence temperature, energy availability, and molecular interactions. These cycles fostered the development of complex systems by encouraging repeated molecular interactions that resulted in the progressive growth of polymers, such as proteins and nucleic acids. Seldom accounted for, periodicity is the force of selection that operated all along. Water plays a crucial role by serving as the solvent that facilitates chemical reactions. Its ability to dissolve solutes, buffer temperature fluctuations, and maintain molecular stability is vital for life's molecular evolution. Water also enables the formation of lipid bilayers, which are crucial for the development of protocells. Carbon-based polymers in the presence of water with periodic alterations in energy flux give rise to the most incredible factor called dynamism. The key to life is dynamism, an inherent capacity for forming, deforming, and reforming. This dynamism involves creating structural states, enabling reversible and repetitive changes. For example, double-stranded DNA can exist as it is, or separate into two single strands depending on environmental factors such as temperature or pH, denaturation. When conditions return to normal, the single strands can reassemble into a double helix, demonstrating reversibility. Proteins also exhibit dynamism with multiple conformations, stabilized by intramolecular non-covalent bonds. These conformations shift in response to environmental conditions, typically reversibly, within a permissible range. Such changes influence the protein's interaction with partners and its functional outcomes, underscoring how dynamic structural transitions are central to molecular function and adaptation. The dynamism of proteins is the key to the functioning of cellular structures and as enzymes. The dynamism opens up the possibility of iterations through which selection for existence operates. Lastly, time is essential for the gradual accumulation of molecular complexity. The billions of years available allowed molecular systems to evolve through repeated iterations, driven by environmental pressures and natural selection, ultimately leading to the primordial replicator. Without time, the slow and incremental process of life's emergence would not have been possible. Urey-Miller experiment, generation of enhanced molecular complexity. The Urey-Miller experiment, conducted in 1953 by Stanley Miller and Harold Urey, demonstrated the potential for organic molecules to form under prebiotic conditions. By simulating Earth's early atmosphere, comprised of gases like methane, ammonia, hydrogen, and water vapor, and applying electrical sparks to mimic lightning, they produced amino acids, the building blocks of proteins. This groundbreaking experiment provided empirical evidence for the abiotic synthesis of life's essential components. While the atmospheric composition used is debated, the experiment remains a pivotal milestone in understanding the origin of life, showing how simple chemicals could give rise to complex organic molecules under primordial Earth-like conditions. Simulating prebiotic selection and evolution modifications in the Urey-Miller experiment, such as the continuous removal of inhibitory molecules and replenishing with more substrates, introduce a drive towards complexity in products. This selection mimics evolutionary processes, albeit at a molecular level, where fit molecules with properties conducive to further reaction or stability in fluctuating environments are enriched. The modified Urey-Miller experiment could yield diverse organic compounds, including precursors to biomolecules like amino acids, nucleotides, and lipids. Successive cycles might enhance molecular complexity, potentially producing short peptides or nucleotide-like structures with primitive catalytic or structural properties. Over time, iterative conditions could foster the emergence of protocellular systems, such as lipid bilayers or microspheres, bridging the gap between chemistry and biology. Selective retention of reactive intermediates might further encourage the formation of molecules with rudimentary catalytic or replicative abilities, offering insights into the earliest steps in life's evolution from simple organic chemistry to prebiotic systems. A modified Urey-Miller setup for organic polymer formation. This modified experiment integrates principles from the original Urey-Miller apparatus with additional factors that promote organic polymers synthesis, including periodic energy input, wet-dry cycles, clay catalysis, selective removal of inhibitory compounds, and substrate replenishment. The goal is to mimic prebiotic conditions that are favorable for forming long chains of nucleic acids and proteins. The gas chamber holds a mixture of prebiotic gases, including methane, CH4, ammonia, NH3, hydrogen, H2, and water vapor, H2O, mimicking Earth's early atmosphere. The reaction chamber introduces energy inputs like electrical discharges and UV radiation, which promote chemical reactions, while the water chamber serves as a reaction medium and a source of evaporation. To enhance polymerization, montmorillonite clay or iron sulfur minerals are added to catalyze reactions, stabilize monomers, and facilitate the formation of nucleotides and amino acids. The system is continuously replenished with prebiotic gases, and byproducts such as CO2 are removed to prevent interference with the polymerization process. Energy inputs are introduced in periodic cycles to simulate natural rhythms. The energy-rich phases involve high temperatures and radiation to activate monomers, while energy-deficient phases involve cooling to promote molecular stability. Wet-dry cycles mimic tidal or seasonal changes, encouraging dehydration synthesis, which is critical for polymerization. A semi-permeable membrane retains reactive intermediates while removing inhibitors to control reaction conditions. Trace ions like Mg-quines and Fe-qupies are also included to stabilize polyanionic molecules like RNA and aid in bond formation, advancing prebiotic chemistry crucial for life's origin. The genesis of the replicator. The origins of life started with the emergence of replicable information. As discussed in the chapter Bioinformation, genetic information is a complex molecule and should be replicable. In the initial stages, there is no need for diversity in the bases, and base sequence does not really matter. The crucial property is replicability, the ability to replicate the nitrogenous base sequence of a nucleic acid strand will make more copies of themselves, leading to the longer existence of that particular molecule with a particular sequence. The first base sequences that have seeded the emergence and existence are those that can replicate, which are self-replicating entities known as replicators. Replicability refers to the molecule's capacity to create additional copies of itself, an important step in the evolution of molecular structures with inheritable information. Replicators can self-replicate, initiating events that allow life to persist, establishing a self-sustaining cycle. Replicators are the first most abundant molecular species that have information. The reasoning is frivolous, for those who replicate will outnumber those who do not. It is like saying that those which can will exist and others will not. The self-replicating nature distinguishes replicators from other molecular entities. It makes them indispensable to the evolutionary trajectory of life on Earth. Model for the emergence of primordial replicators with functional genomes and proteins. The emergence of primordial replicators with functional genomes and proteins is an intricate process that unfolds across several distinct phases. These phases represent critical milestones where molecular complexity, stability, and the ability to preserve and replicate information evolve. Each phase involves iterative cycles of formation and degradation, with the stable complexes outlasting the less stable ones, naturally driving the emergence of life. Phase 1. Formation of complex organic molecules. In the primordial environment, a variety of simple organic molecules, such as amino acids, nucleotides, and fatty acids, form through abiotic processes like those proposed by the Urey-Miller experiment. Conditions like repeating cycles of temperature, pH, and electric discharges lead to the generation of these compounds. Interactions among these molecules, particularly through hydration-dehydration cycles and exposure to varying energy states, facilitate their incorporation into larger structures. These structures may involve RNA molecules capable of folding into stable conformations and amino acids bound by covalent bonds. Through processes like denaturation and renaturation, only the most stable complexes are retained, while less stable compounds degrade. Phase 2. Emergence of chemical polymerization. There is a dynamic relationship between replicability and stability. Anything that is absolutely stable will not deform, and replication requires some degree of deformation, as seen in the denaturation of DNA. Therefore, any entity that is absolutely stable will stagnate, while those that are not stable at all will disintegrate. Molecules with a degree of dynamicity, stable yet conditionally malleable, are likely candidates for replication in the primordial molecules of life. As the chemical environment continues to fluctuate, these small organic molecules undergo polymerization. Through dehydration reactions, amino acids form peptide bonds, and nucleotides form RNA chains. These polymers become increasingly complex as they grow in size and diversity. At this stage, these macromolecules may exhibit rudimentary catalytic activity. The chemical environment selects for these molecular complexes that exhibit increased stability, higher concentrations, and the ability to persist for longer periods. These molecules are more likely to undergo further polymerization, leading to more complex structures with self-sustaining properties. Phase 3. Emergence of enzymatic polymerization. As polymerization continues, molecules evolve the ability to catalyze their own formation, transitioning from mere chemical polymerization to enzymatic polymerization. This step could involve the emergence of ribozymes, RNA molecules capable of catalyzing the formation of peptide bonds or simple protein structures with catalytic abilities. This enzymatic activity dramatically increases the efficiency of polymerization and reduces reliance on environmental conditions. During this phase, homologous recombination may also emerge as a mechanism to repair errors in the polymer chains, facilitating the preservation of information and increasing the accuracy of replication. Phase 4. Emergence of template-dependent polymerization. The next evolutionary leap involves the emergence of template-dependent polymerization, the foundation of modern replication processes. RNA begins to template the synthesis of complementary strands. The base pairing properties of RNA molecules and their ability to catalyze reactions enable the copying of genetic material with increasing fidelity. This allows for the transmission of genetic information from one generation to the next, further increasing the stability and complexity of the molecular structures involved. Over time, these templates allow for the formation of more sophisticated proteins with diverse functions, including enzymatic catalysis, structural roles, and regulatory functions. Self-preservation is the birth of the self. Through these phases, the molecules involved in replication and polymerization undergo selective pressures that favor stability, increased complexity, and self-preservation. The iterative cycles of formation and degradation ensure that only the most stable and functional molecular complexes persist. The self-preserving properties of these molecules, such as the ability to replicate and catalyze reactions, are selected as they increase the likelihood of survival and propagation. Over time, these properties culminate in forming early life forms that can maintain and reproduce themselves. The ability to preserve genetic information and synthesize functional proteins through increasingly complex processes marks the emergence of life. Homologous recombination as a means of existence. As we recognize it today, the emergence of homologous recombination is an extraordinary natural process that likely preceded the need for enzymatic facilitation. It is often studied as a mechanism of repair or generation of variation. Homologous recombination relies on sequence homology and creates new information, a molecule distinct from its two parental contributors. Homology refers to a large-scale similarity in the nitrogenous base sequence of nucleic acids. For homologous recombination to occur, the participating molecules must share significant sequence homology to form hydrogen bonds and stay together. The primordial environment, with its fluctuations in temperature and pH, would have facilitated the denaturation of nucleic acids. Denatured strands naturally renature under favorable conditions, a process that does not require enzymes in such an environment. These iterations of denaturation and renaturation would have occurred cyclically, laying the groundwork for both replication and homologous recombination. Now imagine two homologous double-stranded RNA molecules. Their homology reflects their high sequence similarity, but differences may arise from replication errors or damage. Only the most stable configurations persist during subsequent cycles of denaturation and renaturation. The number of interstrand hydrogen bonds dictates stability. For example, if an unaltered strand from one molecule pairs with its complementary unaltered strand from the other, the resulting hybrid molecule would have the maximum possible hydrogen bonds, making it the most stable. These recombined molecules would persist through repeated cycles, driven by environmental conditions and natural selection favoring stability. The stable ones endure longer in time and dominate in space, increasing their prevalence. Specialized protein machinery may have evolved early during the origin of life. This process not only maintains genetic stability by reversing damage or mismatches, through its elegant simplicity and profound impact, homologous recombination was likely the precursor to more sophisticated processes like repair and sexual reproduction in diverse organisms. Its emergence marked a pivotal moment in the evolution of life's genetic systems by reversing the inevitable changes, the law of reversal of change. We should revisit the role of homologous recombination outside the popular narrative of generation of variation to increase diversity. The genesis of coding, translating base sequence to amino acid sequence. The translation is the change in the language of sequences from base sequence in nucleic acids to amino acid sequence in proteins. The origin of translation is the toughest that I can ever attempt to reason and must necessarily be incomplete. In my view, proto-translation could have arisen from the same pond where replicators have emerged, almost parallel or maybe complementing each other. The difference, though, is that the process of translation emerging is incredibly improbable compared to replicators and could have taken immensely longer. Translation must have evolved first by non-covalent interactions of RNA with amino acids, followed by covalent bonding, charging of RNA, and then followed by polymerization of amino acids into proteins. By this, I am inclined to presume that nucleic acids and proteins have evolved almost simultaneously. These complexes might have increased each other's stability. We must recollect that most of the crucial steps in translation in eukaryotes are still mediated by RNA molecules. Binding of RNA to amino acids. RNA can bind amino acids through specific interactions, primarily facilitated by its unique structure and chemical properties. The nitrogenous bases in RNA can form hydrogen bonds with amino acid functional groups, while the phosphate backbone enables ionic interactions with positively charged amino acids. Additionally, RNA's ability to fold into complex three-dimensional conformations creates binding pockets tailored to specific amino acids, enhancing specificity. A combination of hydrogen bonding stabilizes such complexes, van der Waals forces, and ionic interactions. These RNA amino acid interactions likely played a key role in early biochemistry, enabling the formation of primitive peptide bonds and catalytic activities. In comparison, amino acids wound around by RNA molecules are more stable than free amino acids and free RNA molecules. The reason for this is that the RNA molecules, when wound around amino acids, can form specific binding interactions, increasing the complex's stability. These interactions, such as hydrogen bonding and base pairing, facilitate a more structured and less energetically favorable dissociation. This structure may help protect the amino acids from hydrolysis or other destabilizing processes. In contrast, free amino acids and RNA molecules are more likely to be in an unstructured, free-floating state, which can be more prone to dissociation or other reactions that decrease stability. When amino acids and RNA are in their unbound forms, they lack the stabilizing interactions that could occur when the RNA folds and interacts with the amino acids. This could act as a mechanism of selection for RNA protein complexes. Charging of RNA, the precursor of translation. Assume a soup of RNA molecules, each of which can fold into a conformation that binds to an amino acid. The RNA molecules that fold most efficiently wrap around one specific amino acid. As dehydration progresses or the pH increases, the COOH group of the amino acid becomes deprotonated, leading to increased reactivity. However, I believe it is primarily dehydration that must have played a crucial role in forming amino acid RNA covalent bonds. Dehydration increases the concentration of molecules, and removing water could have facilitated the formation of the covalent bond. A covalent bond between amino acid and RNA would mean more stability than free RNA, and probably also for the amino acid. Formation of peptide bond. Upon several iterations of hydration-dehydration and energy-rich and energy-low cycles, various possibilities could arise. with the formation of a peptide bond between two amino acids becoming more probable. This process would ultimately lead to the formation of peptide bonds. The resulting polymers of amino acids are likely to endure longer due to the strength of the peptide bond, which provides greater stability. Of course, the composition of the amino acids in the polymers would have been of the type of RNA molecules. However, the sequence of the amino acids could be completely random. Template dependency has not yet evolved. Some of the polymers of amino acids or proteins may have acquired properties due to the number of amino acids and the diversity of the amino acids, each with a type of functional group. Some of these proteins could interact with more RNA molecules, amino acids, and other proteins or molecules in the surroundings. Depending on the protein, interacting partners, and surrounding conditions, new properties such as the ability to catalyze reactions are the beginning of proteic enzymes. As enzymes, proteins bind molecules, bend structures, and break bonds through their three-dimensional structures, diverse conformational changes, and diversity of the functional groups. The consequences are in the interacting partners, while the proteic enzymes remain unaltered. Proteins are vital for biological systems and are categorized as structural or enzymatic. Structural proteins like collagen, actin, tubulin, and keratin provide the replicator support, strength, and shape. Enzymatic proteins catalyze reactions, including hydrolases, oxidoreductases, transferases, ligases, and isomerases, driving diverse biochemical processes. The coevolution of nucleic acids and proteins was crucial in forming primordial cells. Initially, RNA molecules likely served dual roles, both as templates for replication and as catalytic entities, forming the first rudimentary enzymes. These RNA molecules could bind specific amino acids, facilitating the formation of early peptides. Over time, the emergence of protein synthesis mechanisms, including ribozymes and peptide bond formation, allowed proteins to take on specialized catalytic roles. The interaction between RNA and proteins in a dynamic feedback loop promoted the development of increasingly complex biochemical processes, ultimately giving rise to primitive, self-sustaining cellular systems with functional genomes and proteins. The dynamism that life represents is the essence and key to life itself. The fundamental basis for the dynamism is molecular conformation, on which every bit of life is manifested. Proteins are one of the best examples of molecular dynamism because of their subunit diversity and length. Their structures are determined by their surroundings and the changes that may happen to them. Changes in conformation change the interacting partners and consequences of the interactions. The consequences determine existence, evolution, or extinction. The sequence of emergence, as depicted in the figure, suggests a progressive development of essential biochemical processes crucial for the origin of life. The sequence is as follows. 1. RNA replication. The first process to emerge is RNA replication, which forms the foundation for the subsequent steps in the evolution of life. RNA, capable of both storing genetic information and catalyzing reactions, likely served as the original template for the replication of genetic material. RNA sequence species are enriched. 2. Translation. Following RNA replication, the next step in the evolutionary timeline is translation. This process involves the conversion of RNA information into proteins, enabling the formation of diverse molecular machines necessary for cellular functions and complexity. Those enriched RNA molecules are likely to form for the protein species, which is likely to have beneficial properties of self-preservation. Number 3, reverse transcription. The emergence of reverse transcription comes after translation. Aided by proteic and ribozymes, this process, where RNA is reverse transcribed into DNA, introduces a crucial step that allows for the integration of genetic material into a more stable form, aiding the evolution of DNA-based genetic information. Reverse transcription may have been a reversible reaction in the sense that DNA could be generated from an RNA template, and RNA could also be generated using a DNA template. While modern reverse transcriptases are largely unidirectional RNA-DNA, a primitive reverse transcriptase in early evolution could have been more versatile, potentially catalyzing both RNA to DNA and DNA to RNA transcription. This would have facilitated early molecular evolution and the gradual emergence of DNA as the primary genetic material. Number 4, DNA replication. DNA replication evolves next. ensuring that genetic material is more stable than all other RNA-based genetic material. RNA primers are still required for DNA replication, indicating a continued dependence on RNA during this phase, further linking the processes of RNA and DNA replication. 5. Transcription. Lastly, specialized transcription emerges. This step involves the synthesis of RNA from DNA, completing the cycle of genetic information transfer, and ensuring the continuous flow of genetic instructions for protein synthesis and cellular activities. From then on, self-preservation has reached dramatic heights because the DNA-based genome is highly stable and, through transcription, can produce numerous RNAs that can be translated to form proteins of immense potential. Evolution of genes, de novo genesis. One of the key factors for selecting a gene that codes for a protein or RNA is its ability to either break a high-energy rich molecule to yield energy or eliminate, degrade an inhibitory molecule. Secondly, the consequence of the interaction should confer an advantage to the encoding entity. Therefore, the first proteins that would be selected are those whose substrates are high in concentration. In the prebiotic era, carbohydrates likely were the most prevalent of all molecules because their probability of formation was high. This is attributed to the fewer types of elements required for their formation, the abundance of these elements, their reactivity, their ability to form under energy-providing conditions, stability, and importantly, their water solubility. Thus, the first proteins selected would likely have been those capable of harvesting energy and matter from these molecules to build more of the entity that encoded them. It is also possible that the energy trapped in carbohydrates may have driven the entire process of abiogenesis. An identity needs to have a high number of representatives, entities. For an entity to achieve this, replication is a fundamental requirement. The replication of a protein encoding gene must be associated with a propagating entity, genome. Therefore, irrespective of what may have preceded abiogenesis, life begins with the emergence of the replicator. The de novo genesis of genes in the prebiotic era represents a fascinating interplay of molecular interactions and natural selection. Proteins that emerged during this time through random polymerization of amino acids inevitably interacted with various molecules in their environment. These molecules could include their kind, other proteins, lipids, carbohydrates, nucleic acids, DNA or RNA, water, ions, or other chemical entities. Such interactions could be covalent or non-covalent, each with distinct consequences. Covalent interactions often lead to more complex and stable structures capable of mediating processes beneficial to the encoding molecule. These structures could facilitate catalytic activities, structural stability, or other functions valuable for self-preservation and replication. On the other hand, non-covalent interactions, characterized by hydrogen bonds, ionic interactions, or hydrophobic effects, could transiently alter the protein itself or the molecules with which it interacted. These modifications could involve degradation, addition of functional groups, or reorganization of existing molecular frameworks. The outcomes of these alterations were diverse, ranging from increased structural or functional efficiency to complete loss of activity. Crucially, among the myriad interactions and modifications, only those proteins that conferred an advantage in self-preservation, replication, or other survival-oriented processes were passively selected for. This selection was not active, but a natural consequence of stability and utility. Proteins encoding interactions that increased fitness by enhancing survival, replication rates, or adaptability gained an edge over others. Variance, an inevitable parameter in such a stochastic environment, introduced diversity, enabling some molecules to outperform others in evolutionary success. This process of selective advantage laid the foundation for the emergence of new genes de novo. The proteins with beneficial consequences encoded and preserved their structural or functional properties. Over time, such encoding led to stabilizing and evolving specific genetic sequences that supported advantageous protein structures and interactions. While the first of the genes were the replicators, they must have been followed by the emergence of genes that could harvest energy from the surroundings. The de novo emergence of genes, I think, is proportional to the molecules that are energy-rich and abundant. The same principles of de novo genesis persist even in modern cells, where novel genes arise through interactions and natural selection driven by molecular and evolutionary forces. This highlights the continuity of fundamental principles from the prebiotic era to contemporary cellular biology. In the contemporary cells, the scenarios are more streamlined and directed. The first requirement for a novel gene is its expression. Suppose the gene product is non-toxic and does not interfere with essential cellular processes. In that case, it has a chance to be retained within the genome. Over many iterations of replication and mutation, these gene products may undergo further modifications, improving their functionality. For example, a protein initially interacting weakly with a metabolite might evolve a specific binding site, enhancing its utility within the cell. Selection pressures play a subtle but significant role. While early-stage novel genes may not immediately confer a direct advantage, they persist through neutral selection simply by being non-detrimental. Over time, slight advantages conferred by improved protein functionality can be indirectly selected. For instance, a protein stabilizing cellular membranes by interacting with lipids or assisting in basic metabolic processes by binding ions might enhance cellular fitness. As these advantages accumulate, the gene becomes more indispensable, transitioning from neutral to adaptive. The interactions of novel proteins with their molecular partners underpin their evolutionary significance. Binding to nucleic acids can facilitate regulatory roles, such as influencing gene expression or stabilizing RNA molecules. Protein-protein interactions enable the assembly of larger complexes, driving cellular organization and signaling. Interactions with ions, cofactors, or metabolites can enhance enzymatic activity, directly influencing metabolic pathways. Hydration, mediated by water molecules, stabilizes protein folding and solubility, further promoting functionality. Crucially, the structural and functional diversity of proteins enables them to explore a vast molecular landscape. This versatility increases the probability that a novel protein will find a functional niche. Over evolutionary time, such proteins may develop specialized roles, becoming integral to the organism's survival and reproduction. The gradual optimization of these interactions, combined with the selective retention of beneficial traits, allows for the stepwise evolution of complex gene products, ultimately contributing to the sophistication of modern genomes. Evolution of repair mechanisms. The whole universe works to exist in the backdrop of an immense landscape of change. This is more so for living things because they have higher thermodynamic states and also a possibility of environmental stringency. Mistakes will happen spontaneously and naturally. For existence to be manifested, each entity should work towards repairing the damages or variations to the genetic information. Therefore, every cellular entity that we know has diverse repair mechanisms. Exploring repair mechanisms in the context of evolution uncovers a fascinating journey within the complex dance of life. Repair mechanisms are organisms' biological responses and processes to fix damage or errors in their genetic material, cellular structures, or physiological functions. To understand the evolution of these mechanisms, we must delve into why they exist and the need for variation in living systems. From simple single-celled organisms to complex multicellular entities, intricate systems have evolved to protect genetic information and cellular structures. Preserving the accuracy of genetic material is crucial because errors or damage can affect the viability and functionality of an organism. Repair mechanisms have evolved as a strategic response to environmental factors, radiation, chemical exposures, genetic replication, and cellular processes. Evolution is an ongoing process of adaptation and selection. Living organisms navigate a dynamic environment where genetic diversity is crucial for their ability to adapt and thrive. Repair mechanisms act as guardians of genetic fidelity, promptly correcting errors during DNA replication or caused by external factors. This pursuit of genetic accuracy aligns with the overarching goal of evolution, enhancing an organism's chances of survival and reproduction in a competitive and unpredictable world. Variation at both genetic and phenotypic levels is essential for evolutionary success. Repair mechanisms contribute to maintaining genetic diversity by preserving the integrity of the genetic code. However, this raises an intriguing paradox. While variation is inevitable, The existence of repair mechanisms implies a counterbalance, a way to correct errors and maintain stability. This delicate equilibrium between variability and stability reflects the sophisticated nature of evolutionary strategies. Repair mechanisms are deeply intertwined with the fundamental principles of natural selection. Organisms with effective repair mechanisms can survive and pass on their genetic material better. This selective advantage leads to the prevalence of individuals and species with robust repair systems. The evolution of repair mechanisms is evidence of life's persistent drive to adapt and perpetuate its existence in the face of environmental challenges.